Good day, class. Today we're going to be doing the immunofluorescence assay. Introduction. Immunofluorescence is a technique that utilizes fluorescent labeled antibodies to detect specific target antigens. This technique is used in the detection of autoimmune diseases. An autoimmune disease is one in which the body produces antibodies that attack its own tissues and can be separated into two categories. Those that are categorized by the presence of autoantibodies that are broadly reactive with nuclear or cytoplasmic antigens and do not demonstrate tissue specificity, such as anti-nuclear antigens, ANA. And those that are characterized by the presence of autoantibodies which demonstrate tissue specificity, such as double-stranded DNA, also known as dsDNA. Antibodies that bind to DNA belong to the group of anti-nuclear antibodies, ANA. A positive ANA test may indicate systemic lupus erythematosus, SLE, mixed connective tissue disease, scleroderma, and Sjögren's syndrome, dermatomyositis, polymyositis. A positive double-stranded DNA, dsDNA test, however, confirms systemic lupus erythematosus. So we're going through with the procedure. In the case of ANA and dsDNA, ANA, the patient's serum has to be diluted 1:40. So an example of that is 25 microliters of the serum is added to 975 microliters of PBS. And for dsDNA, it's a 1:10 sample dilution. An example of that is 100 microliters of serum is added to 900 microliters of PBS. Here we have the two slides to do the immunofluorescence. The very first one, the upper to you, will be the ANA, which has a positive and a negative represented by a triangle, and we have positions 1 to 10 where our samples will be placed. For DNA, we have a positive and negative represented by a square, and samples 1 to 4 are placed in the circle. So after we've created the dilutions for either the ANA and the dsDNA, we are now going to add the sample into the respective positions. So class, I'm now going to demonstrate the actual addition of our control. In the case of the ANA, it's represented by a triangle. So I'm going to add the 20 microliters of the control. So once it has been placed, we are now going to follow the contours of the actual triangle. In the case of the control for the dsDNA, it's represented by a square. So once again, I'm going to add the control, positive control in this instance. Then I'm going to take the tip and run it to the edge. In the case of the actual samples in both ANA and dsDNA, they're circular. So I'm going to add in position 1 for the ANA. And once again, I'm going to take the tip and follow the outline. Once the samples are placed in your controls and the wells, we now have to place the slides into a moist chamber. So this is what we need in order to create a moist chamber. So we're utilizing it has, this is what we use in micro B, but in this instance we're going to use it as a moist chamber. So we're creating, we take the sticks, place the tape so that it won't roll around. We place cotton ball to create the chamber, and we place our slides and we cover. That's the moist chamber. So this has to stay for 30 minutes. So class, after taking it out of the moist chamber, you're now going to be washing your slide. So I want to put some emphasis on the ANA slide itself. The positive is to the top, so you need to tilt the positive away from the others. And the technique when holding your pipette between your fingers, you squeeze, you place it in, and you run each well, you tap, you tilt it the other way, you tap, 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 the excess. So after you've done that class, we're going to put it in your PBS. And once we put it in here, we need to agitate on entry. And in the middle of the 10-minute session and at the end. So it stays in this for 10 minutes. Once the 10 minutes is up, we remove the slide. You'll have distilled water. Ensure that you cover each of these wells. That is to remove any residual PBS. Now going to learn to wipe the excess up. Now it's a technique. You have to ensure that you are not wiping away the actual slide itself. So one indicator is the positive, the negatives, and the numbers are facing you. And if we look at it this way, we're going to see that is the reverse. So definitely we need to have the numbers and the signs up. So we'll be wiping beneath. We wipe the edges. So after you've wiped the edges, the top and the edges, we place it back in the moist chamber. Remember class, we don't want this particular slide, the ANA and the dsDNA, to dry out at all. So once we've wiped the edges, we're now going to be adding the fluorescent goat anti-human. So place it in the moist chamber, and we're adding 10 to 15 drops. 30 minutes. 30 minutes is up with the FITC. Going to put it back in the PBS for 10 minutes. Agitate on entry. In the middle and at the end. Once the 10 minutes is up, we're going to place it in the distilled water. Make sure you cover the entire surface to remove the excess PBS. Once again, the bottom, the edges, ensuring that the signs and the numbers are on top so you're not wiping away the slide itself. You place it back in the PBS. To put the counter stain. The counter stain is Evans blue. Agitate. Final step before we use the microscope. So we're going to take it out from the counter stain. Same technique again, underneath, sides. So we're going to use the buffered glycerol, which is the media between the slide and the cover slip. Put 3 drops. The cover slip as close as you can. What we're trying to do, class, is to avoid bubbles. So now we can take our slide and use a fluorescence microscope using the 40x objective. So we have the interpretation of the results. In the case of the ANA, a positive is, the serum is considered positive when the nuclei fluoresce brighter than the negative control. So it is very important that, yes, you use both positive and negative, but the interpretation of the results is based on brighter than the negative control. Well, with a clearly discerning pattern of fluorescence. So in the ANA, you have the different types of fluorescent patterns. You have homogeneous or diffuse, where the whole nucleus evenly stained. Peripheral or rim, where the periphery of nucleus fluoresces most brightly. Speckled, the nuclei of the cells have a greeny appearance. Nucleolar, the nuclei stain. Discrete speckled, nuclei contain many discrete speckles that are centromere, distinct, and interrupted throughout the nucleus. The degree of the fluorescence may be semi-quantitated by the following: 4x maximal fluorescence, 4 plus maximal fluorescence, 3 plus less brilliant yellow-green fluorescence, 2 plus definite cell pattern but dim fluorescence, 1 plus very subdued fluorescence. A serum is considered negative if the nuclei of the cell fluoresce with about the same degree of brightness as the negative control and lack a clearly discernible pattern of fluorescence. In the case of the dsDNA, for interpretation of results, only cells with a well-defined morphology should be read in each field. View a positive control well prior to reading each patient's serum well to become acquainted with the orientation of the cells. In a typical cell, the kinetoplast will be located towards the basal body and posterior to the nucleus. Interpretation. A positive test for anti-DNA is observed as a brightly yellow-stained kinetoplast. This structure is somewhat smaller than the nucleus and is often located near the periphery of the organism. Nuclear staining may be observed in some instances also, since sera may contain antibodies directed against other nuclear antigens in addition to DNA. Kinetoplast staining can be differentiated from nuclear staining on the basis of the smaller size, brighter stain, and peripheral location of the kinetoplast. Two additional staining patterns which are occasionally observed are as follows. The organism is stained nearly to the insertion of the flagellum or at the region of the basal body. This staining is of certain significance. There is perikinetoplast staining with a total absence of staining of kinetoplast itself, in which appears as a whole. This pattern is also of uncertain significance and should be considered negative for antibodies to DNA. Recording intensity of fluorescence. Kinetoplast. Positive control, 4 plus, strong, negative control, plus minus, red orange. Fluorescence just visible, 1 plus. Fluorescence half that of the positive control, 2 plus. Types of fluorescence observed and interpretation. No kinetoplast, no nuclear fluorescence, negative. No kinetoplast, positive nuclear fluorescence, positive for anti-nDNA. Positive kinetoplast and positive nuclear fluorescence, positive for anti-nDNA. Positive fluorescence staining of the basal body only, non-specific reaction, negative for anti-nDNA.